To your attention, a report on the topic Ecological determinants of algal communities of different types of ecosystems. Forest and herbaceous ecosystems have different abilities to influence the formation of their internal phytocyanotic environment and the composition of the organisms that inhabit them. Various studies have shown the specific features of algal communities in forest, steppe, desert, and other ecosystems. The analysis was carried out on the basis of material collected in 2000-2013 in the south and southeast of Ukraine in oak, alder, aspen birch, and pine forest ecosystems, in floodplains of Samara, Seversky Donets, Vovcha, Molochny rivers, in oak forest ecosystems in gullies, in the steppe and saline in flat habitats. Algae of various divisions were found within the studied ecosystems. A wide variety of chlorophyte and heteroconphyte species are noted in forest ecosystems. They account for 39.7%, 59.7%, and 28.9%, 45.3%, respectively, of the total number of algae species in forests. The majority of heteroconphyte is represented by species from the classes Xanthophyceae, Bacillariophyceae, 17.5%, 27.4%, and 7.9%, 16.9%. The diversity of Eustigmatophyceae from heteroconphyte, streptophyte, and Euglenozoa is low and limited to a few species. Cyanobacteria in forest algal communities range from 2.7% to 15.7% of the total number of species. Thus, within each ecosystem, a specific ecological space is formed, which is assimilated by various taxa of microalgae. Using principal component analysis, 4 main factors with eigenvalues greater than 1 were identified, which explains 81.4% of the total variance. When calculating the factor loadings matrix, the rotation of factors was used by the Verimax normalized method. The first factor, which explains 34.82% of the dispersion, is associated with the type of ecosystems and such edaphic parameters as pH, the availability of moisture in the habitat, and soil mineralization. Along the first axis, on one side, there is a transition from swampy to xerophytic steppe ecosystems, from ecosystems without sign of salinity with weakly acidic pH to saline ones with alkaline pH values. The second factor reflects changes in the gradients of trophicity, humus content, and particle size distribution of soils. It accounts for 21.98% of the total dispersion. Along the second axis, there is a transition from ecosystems with low humus soils of a lighter particle size composition to ecosystems with highly humified soils of a heavier particle size composition. The third and fourth factors additionally explain 16.04% and 9.27% of the total data dispersion, respectively. Their values mainly dispersion on the mineralization, trophicity and moisture supply of edephotopes. Among the significant factors affecting soil algae, pH, organic matter, particle size distribution, and moisture content were also indicated. In terms of composition of microalgal communities, the first main component is indicated by the diversity of cyanobacteria species, the second is chlorophyte, streptophyte, eustigmatophyceae from heteroconphyte, the third is xanthophyceae and euglenozoa, and the fourth is bacillariophyceae. For cyanobacteria, the most significant characteristics of the edaphotope were found to be pH, mineralization, providing moisture, and the type of ecosystem. Green algae, chlorophyte, streptophyte, and eustigmatophyceae exhibit similar requirements for the ecological parameters of their habitat, such edaphotope characteristics as trophicity and particle size distribution are of particular importance for them. The provision of the habitat with moisture is of particular importance for xanthophyceae and euglenozoa, and for bacillariophyceae, the trophicity and mineralization of the edaphotope. Thus, in the course of the research, the specific richness of algae in each ecosystem was established, and a multidimensional ordination of algal communities was carried out based on the analysis of the main components to clarify the factors that determine the composition of algal communities. Dear colleagues, thank you for your attention.